Hey, we're doing chapter 15. Chapter 15 is on sensory. So what you're looking at here is the anatomy of the eye. Just to go over and highlight some areas. The opening of the eye that allows light into the eye is referred to as the pupil. The part of the eye that is smooth muscle that controls the diameter of the pupil to allow a certain amount of light into the eye. It's the actual color portion of the eye. It's a smooth muscle. It's referred to as the iris. And then the white portion of the eye here is referred to as the sclera. Okay, let me clear this out. So the drawing here, just to go over those areas again, um, they don't show the people in this particular picture, so not labeled at least. So this would be the pupil. Okay. Um, and then the transparent part of the eye here is called the cornea. Transparent means it's going to allow the light to go through. And then again, the white portion of the eye here is referred to as sclera. You have some little infoldings of the cornea called conjunctival sacs. And you can see on the sclera, that's where the skeletal muscle is going to be attached. That allows you to move your eye. So here you're looking at the lacrimal gland up above. Lacrimal gland produces tears. It's going to help lubricate the eye. Also, you have some antibacterial proteins in your tears. So hopefully nothing takes housing on your eye. And times of stress, the theory is you release toxins also as well in your tears. The tears will eventually enter the lacrimal sac, right? It enters the sac through this little canal called the lacrimal canaliculus, which can drain eventually into your nasal cavity. These are the muscles involved in moving your eye. So let's go to a larger picture here. Muscles that have the name rectus in the name of the muscle, the way it's named is the way it moves the eye. So superior rectus muscle move the eye above, lateral rectus muscle move the eye lateral to the side. However, if you got a oblique name in the muscle, it's actually moving in the opposite direction. So like superior oblique actually moves the eye down and then to the side. So let's try to draw a little down arrow. Whereas where is it? The inferior oblique will actually move the eye up. Okay. So again, if the muscle has rectus in its name, it moves the eye in that direction. If it has oblique in its name, it moves it in the opposite direction. You also have an inferior rectus muscle that'll move the eye down. So let me reiterate what I just said. Anytime you have rectus in the name of a muscle, that's the way it's going to move the eye. So lateral rectus will move the eye laterally. Medial rectus will move the eye midline, medially. Superior rectus would raise the eye. Inferior rectus will lower it. However, if you have oblique in the name of the muscle, it moves in the opposite direction. So you'll notice inferior oblique will actually move the eye up, whereas superior oblique move the eye down. Most of these muscles are being communicated by cranial nerve numeral 3, which is called oculomotor. The other two nerves that are involved in communicating the muscles moving the eye is cranial nerve 4, trochlear, and cranial nerve 5, which is called abducens, which would move the eye to the side like abduction. And this is just an alternate view of the muscles that move the eye as you're looking at it from a superior view. Okay. Oh, just to mention, cranial nerve 4 communicates to the superior oblique muscle. So your superior oblique muscle actually moves the eye down. It's attached to the eye through a pulley, which pulley referred to as trochlea. So cranial nerve numeral 4, called trochlear nerve, communicates to this muscle, therefore pulls the eye down to the side. Yes, we saw this picture already. Some of the anatomy of the eye. Let's go over this real quick. So the transparent part of the eye again is called the cornea. Transparent means it's going to allow all the light to go through. Something that you could see through like glass is transparent. Okay. It's not the same as translucent. Translucent, think of something that allows most light to go through, but you can't quite make the image out on the other side of the material. Think of like frosted glass or a shower curtain. So the cornea is actually transparent. There are segments within the eye, or you could refer like cavities, but they're called segments. You have a anterior segment, and the fluid in there is referred to as aqueous humor. And then all back here is referred to as the posterior segment, and the fluid is referred to as vitreous humor. Now, the fluid is produced by capillaries located within the eye. It's not, can't see here, but the capillaries produce the fluid. And then eventually it drains into a large thin wall vein called a sinus to re-enter your bloodstream. So light goes through the opening of the eye. The opening again is called the pupil, and the pupil is controlled by smooth muscle, referred to as your iris, which you know as the color portion of your eye. But it is a smooth muscle that controls the diameter of the pupil. And then you have this structure, it's called the lens, and the lens helps to focus light on a specific area where you have only cones. Cones are a type of photoreceptor to interpret color and visual sharpness. So when you get to that specific area called the fovea centralis, that's an area where you only have cones where the lens tries to focus light upon so that you can focus on objects either near or far. So that is the job of our lens, is to give us the ability to focus on objects either far or near. Now, how does the lens do this focusing on objects near or far? It pretty much changes its shape. Okay. We'll look at that in slides later. But what controls the shape of the lens is smooth muscle here, referred to as the ciliary body. And they're attached to the lens through ligaments here, known as suspensory ligaments. So when the ciliary body, this smooth muscle, when it contracts, it actually, the ligaments, they go flaccid or loose, allowing the lens to bulge more. And when you do that, you tend to focus on objects near. And then when the ciliary muscle, or we say ciliary body, relaxes, they're pulling on the ligaments, therefore stretching out the lens, therefore giving you the ability to focus on objects far. So light travels, has to get past the cornea, which is transparent, goes to the pupil, and then the lens will focus light on a specific area where there's nothing but cones called the fovea centralis. Now you have two types of photoreceptors to interpret what you're seeing. You have what are called rods that tend to amplify light when there's very little. So you use rods in night. And then cones think of like, for me, I think crayons because they're cone shaped. The cones, those photoreceptors interpret color and they give us visual sharpness. So in this structure here, and drawn in yellow, this is called the retina. And this is where you find both photoreceptors, rods and cones. And overall, we have more rods. However, when you get to this little circular region here, that whole circular region is called the macula lutea. Within the macula lutea, you actually have more cones. And then right in the middle of the macula lutea, called fovea centralis, That's where you only have cones. But again, overall, you have both photoreceptors in the retina. Don't forget, you have more rods than you do cones. Yet when you get to this circular region called macula lutea, it shifts. You have more cones than you do rods. And then in the middle, called the fovea centralis, there's nothing but cones. And that's where the lens tends to want to focus light on, so you can focus on objects near as well as far. And then behind the retina, you have what's called the choroid, and you're going to see blood vessels distributed out the choroid, and that's going to provide oxygen, nutrients to the photoreceptors, as well as pick up their waste products. And then the area here, often known as your blind spot, okay, it's called the optic disc. That's the appropriate term, but you could see they can often sometimes refer to it as your blind spot. The reason it's considered a blind spot is because there's no photoreceptors there. There's no rods nor cones. Actually, what you have here are axons of neurons that form cranial nerve numeral 2, which is your optic nerve. It's going to send information to the brain to interpret what you're seeing. So in here in the optic nerve, there's a blind spot called the optic disc where there's no photoreceptors. And as well, in this area, you're going to allow blood vessels to enter as well as to exit the eye. An actual picture here of a human eye that's been freshly dissected. When you have time in lab, if you get to see a bovine or sheep eye, it won't look as fresh because it's been sitting in preservative. So again, some of the areas tend to highlight here. The transparent part, it'd be the cornea. And just to take note, if you do see a dissected eye, it's not going to be transparent any longer. That preservative is going to make it cloudy. The opening of the eye is the pupil. This here is the lens. The anterior segment here, where you have aqueous fluid, and the posterior segment, where you have the fluid called vitreous humor. And the white portion of the eye, where muscles as well as fats are attached to the eye, that's called the sclera. And yeah, around the eye, toward the back, you have lipids in an attempt to conserve heat so you don't lose heat from the body. So if you get to see a dissected eye, you may see a lot of lipids around the eye. And then this area here, the blind spot known as the optic disc, is where you form your optic nerve. And what controls the diameter of the pupil? Again, it's the smooth muscle, which you know is your color portion of the eye. It's called the iris. Transparent part of the eye up front, the cornea. What allows you to focus on objects near and far? Again, that's called your lens. So this is a picture just showing us that the smooth muscle of the eye, called the iris, again, that you know is the color portion, controls the diameter of the pupil to allow a certain amount of light in. So we talked about the two types of divisions, the autonomic, which you have sympathetic and parasympathetic. They do communicate to the smooth muscle of the eye, right? So during fight or flight, which is your sympathetic, you want to see all your surroundings in a threatening or physical activity. So the iris, when it's communicated by sympathetic, will cause the pupil to dilate. Vice versa in parasympathetic. Parasympathetic is a time of doing digestion, relaxation, conserving energy. You're going to see the portion of the iris start to constrict the pupil so you don't allow as much light into the eye during parasympathetic activity. Okay, let's see if we get a larger picture here of each little section. And let me get the marker out. Okay, so here again, we're looking at the optic nerve, right? These here, you got axons of neurons that are going to send information to the brain to interpret what you're seeing. And the photoreceptors that get activated, they're located within a structure called the retina. Okay, so in the next picture, you're probably going to see this enlarged. So this is the way light travels. It actually goes past the cornea, past the anterior segment into the pupil, then gets directed by the lens to the fovea centralis, where there's nothing but cones. So typically to stimulate the photoreceptors, light stimulating them from the back. So that means all this area has to be transparent to allow light to stimulate the photoreceptors. So this is the picture of the photoreceptors where I said they would enlarge. The next slide. And let's get the marker out. So in purple here, these are the rods. Rods, again, amplify light when there's very little. So most often, yeah, you're going to use them at night, right? And then in less number, you have the cones, and you can see that they're cone shaped. Cone's job is to interpret the colors, the different colors we see, and to give us visual sharpness. So when light stimulates these photoreceptors from the back, they'll send signals to other cells here, some bipolar neurons, eventually to some ganglion neurons. And it's the axons of the ganglion neurons that eventually form your optic nerve. And one thing to take note here is light has to stimulate them, and light's coming actually from the back. So light has to be allowed to go through to stimulate the photoreceptors. This is an actual stained tissue the eye put under, a microscope. And the little dots that you see are going to be the cell nuclei of the different neurons. So let's have a look here. So here are the nucleus of the photoreceptors, both the rods and the cones, whereas the little dots here are the nucleus of the bipolar cells. And then eventually they send signals to the cells. Here we can see the nucleus of the ganglion cells. And eventually the axons of the ganglion cells again form the optic nerve. In front, or you could say in toward the very outermost region here, this area is called where is it? The choroid. And it's the choroid where for us humans, it's dark, it's black, okay? And just to let you know that things that give the appearance of black, they don't refract light, they absorb it. So any light that doesn't stimulate the photoreceptors here will get absorbed by the choroid. Other organisms that see very well in the dark, they're going to have here a choroid where it look more like teal in color instead of black. So that way, if light misses the photoreceptors, it gets refracted, bent back onto the photoreceptors to stimulate them again. So organisms, they have a lighter colored choroid so they could see better at night. So if you just take note of that when you look at the sheep eye or bovine eye, these are organisms that see a little bit better at night. So you'll see a different colored choroid, more teal in color. This is just looking at a picture. You may go to your optometrist. They'll have a look at your eye and everything. So they see the blood vessels, and you can see the area where your optic nerve forms, known as your blind spot. It's called the optic disc. And again, an area where you want to focus light, where there's more cones. It's called the macula lutea. And then right in the middle, it's called the fovea centralis, where you have nothing but cones. I kind of relate this to a dartboard, where you get to a dartboard, there's more rods within the dartboard than cones. I mean, sorry, more cones overall now on the dartboard than rods. And then when you get to the bullseye, called the fovea centralis, there's nothing but cones. All right, let's clear all this out. So here we're looking at a drawing of the eye again. Just to reiterate certain things we need you to know. The transparent part of the eye, called the cornea. The front segment here of the eye, it's called the anterior segment that has fluid, referred to as aqueous humor. The posterior segment behind the lens has fluid as well, but it's referred to as vitreous humor. And again, the fluid's going to be made by capillaries. Here you can see the fluid being produced by indicating there's capillaries here in the ciliary body, which is smooth muscle. And then the fluid forms the fluid in the back segment, also the front, and then it gets recirculated into your bloodstream through a large thin wall vein called a sinus. The other thing your book will bring to your attention is that there are no capillaries in the cornea. So theoretically, you can get a cornea transplant. Your body wouldn't reject it since you don't have capillaries there. Would it not be recognized as foreign? And then here you could see the lens, which job is to do what's called accommodation, which gives us the ability to focus on objects near or far. And what controls the shape of the lens is the smooth muscle here called the ciliary body, and it's attached to the lens through ligaments called suspensory ligaments. And the lens, as we get older, think of like an onion, you start to add more layers as you get older. So what you were able to do when you were younger, which is to focus very well on objects near or far, as you start adding more layers, it's going to be more challenging, more difficult to focus on objects near or far as you add layers to your lens. Oh, this is showing cataracts where the lens hardens. Some people may be genetically prone to that, where proteins in the lens start to harden. You'll often see this like in diabetics where they have too much glucose in their bloodstream, causing damage to the capillaries that would provide oxygen and nutrients to cells. So if you got damage to capillaries, tissues tend to die, and in this case, the tissue here, the lens would harden. So that's cataracts. Now, this is showing that how we interpret color, well, some forms of energy or a lot of forms of energy, they travel in waves, okay? So you're going to see here in this picture that certain types of energy can travel in waves. You've heard of X-rays, right? UV rays, gamma rays, right? Radio waves. And waves, if they have a high frequency, so high frequency is how many times something occurs per time. So I'm drawing a wave here. You can see it occurs 1, 2, 3, 4, 5 times. And let's say this wave only occurs 2 times. So this wave here that I drew has a higher frequency. So waves that have a very high frequency tend to be damaging to our cell's nucleus that can destroy the information for growth or repair. It has all the instructions for proteins, right? So you don't want too much overexposure to UV rays that may cause damage to your cell's nucleus, maybe causing cancer, nor do you want overexposure to X-rays because they have a high frequency. Radio waves, they have a very long frequency, or short, I should say. In other words, a radio wave travels a good distance before it forms another wave, which would not be as damaging to waves that travel in higher frequencies. Okay, so what are we looking at here? You're looking at white light or visible light. You probably notice after a rainy day when there's still moisture in the air, you'll see the formation of a rainbow. So technically, you could shine visible light through a spectrum, it'd be separated out into these individual wavelengths of color. Now, how do we interpret color? Typically, if you have a material, whatever wavelengths being bent back to the observer, that's the color that you interpret that you're seeing. So let's say a material appears to be blue, that material's bending back to you, the wavelength in blue. Where did the other wavelengths go? Well, if you're not seeing them, they're being absorbed by the material. If I see a particular material and it looks red, well, that's the particular wavelength that's being bent back to me, the observer, and the other wavelengths are being absorbed. The only exceptions is black and white. A material that will absorb all the different wavelengths and not bend back any of them to you, that gives the material the appearance of being black. So typically, black's not really a color, it's just a material that absorbs all the wavelengths of light. Vice versa, if something appears white, all the wavelengths of this color range are being bent back to you the observer, and you interpret it, the material as being white. Okay, let me close this out and go to the next picture. So again, wavelengths with very high frequencies are damaging to our cells like X-rays or overexposure to UV rays versus waves that have a low frequency like radio waves typically are not harmful. Okay, can light bend? Yes, you could see here in the picture that obviously we know by experience that the spoon is not bent, right? So light, when it's traveling in one atmosphere, is traveling at an angle, but when it hits a different medium, bends, and then it's being bent back to the observer at a different angle. Therefore, the material in the water looks bent because light can bend. Okay, so like I mentioned earlier, as we get older, we tend to have a harder time focusing on objects near or far. People that are nearsighted can see things fine near, but have trouble seeing far. Vice versa, people that are farsighted see the images fine far, but have trouble seeing things near. So we can correct that with lenses. I'm not going to go in detail, just know that lenses can correct if you have any problems doing accommodation, which is the ability to focus on objects near or far. Right. So, and what part of the eye does accommodation again is your lens. Okay. So here the lens is going to be stretched when you're focusing on objects near. I'm sorry, far, not near. And what part of the autonomic system does that is the sympathetic. Okay. And notice that the image would fall inverted onto the fovea centralis, yet your brain's going to interpret it as right side up. And vice versa, when you focus on objects near, okay, so what's in control there is parasympathetic activity, and then the lens is going to bulge to allow you to focus on objects near. And then here they're showing these the suspensory ligaments that the lens is going to be attached to, and then the muscle called ciliary muscle or body that controls the diameter or shape of the lens. A normal shaped eye is referred to as emmetropic eye, where you do have the ability to focus objects just fine onto the fovea centralis. Now, astigmatism. There's a term called astigmatism, which means you don't have the ability quite well to focus on objects near or far. And that can just occur over time as you age, you start adding layers to your lens, it's going to be harder to focus on objects. However, you may be born with an irregular shaped eye that it can also cause astigmatism. So a person, they're showing us here in the drawing with what's called a myopic eye. They are nearsighted, which means they can focus on objects near, but they have problems focusing on objects further away. And here you have an irregular shaped eye where it's actually a little bit longer than normal. Okay. So the image falls a little short of where it should go. And that can be corrected by lenses. In this case, it says a concave lens where it kind of curves in here. And then let's say people with a hyperopic eye, these people are farsighted, which means they see images fine far, but have problems seeing images near because they have an irregular shaped eye where it's not as long as it should be, and the image falls further away than it should. And this can be corrected, it says here, through convex lenses. Convex means curves out. Now, you probably heard of LASIK surgery. Basically, in LASIK surgery, they somehow, I'm not the specialist, they reshape not the lens, but the cornea so that it bends, light bends correctly off the lens to the area that it needs to go. Okay, so in this picture, we're looking at the rods and cones. Let me go to a larger picture here. Again, overall, we have more rods, and rods are to amplify light when there's very little. So we use them at night, okay? And you'll notice that light is stimulating the photoreceptors from the back. And then what we have in less amount are what's called cones. They're cone shape. This helps us to interpret what color we're seeing and to give us visual sharpness. And the cell of the photoreceptors, there's two parts. There's an outer segment and an inner segment. And this outer segment, the outermost region is constantly having friction occur. So they are constantly having to reform new parts of the outer segment. Somewhere in one of these slides, we're going to see that. Here we go. I think it's illustrated better here. So in this outer segment of the rods and cones, you have a cell membrane shaped in kind of like in stacks on top of each other, similar like if you would stack pennies, where the pennies represent the cell membrane. And in the cell membrane, you have proteins here that have a portion of the protein called opsin. Okay, so that's going to absorb light. All right. And when it absorbs light, it's going to initiate eventually the signals to interpret what you're seeing. Okay, so the pigmented portion is called opsin, and then the other portion of the protein is called, where does it say? Oh, I'm sorry. The pigmented portion is the retinal, and then the protein portion is the opsin. Okay, together it's called rhodopsin. Okay, so this is a better picture. Within these penny-shaped stacks of the cell membrane, you have the pigment part that absorbs light. It's called retinal. And then you have the protein part. The rest of it is opsin. And together it's referred to as rhodopsin here. If I can highlight it. Okay. And when it comes to cones, you basically have three types of cones. In other words, they have three different types of retinal. You have cones to interpret, I believe, red, blue, and green. And just the way you stimulate the three types of cones will give you the interpretation of different colors. Okay, so here the pigmented portion again is called retinal. And you're going to notice that when light hits the retinal, it's going to change its shape. Here it's bent. The structure of the protein called retinal, the ending part looks bent, but when light hits it, it changes its bent structure to a straight structure. So these are referred to as isomers. It's basically the same chemical, same molecule. In other words, you have the same amount of hydrogen, same amount of carbons, and same amount of oxygens. It's just arranged a little bit differently. So when light hits 11 cis retinal, it changes into an isomer form called all trans retinal. And that initiates the signaling in the photoreceptor to eventually make action potentials that will send signals to the brain to interpret what you're seeing. And once that starts the process, that all trans retinal does have to go back and reshape itself in the choroid back into the 11 cis isomer form. Okay, so here we got the light. The light when it goes ahead and hits the retinal, this is the retinal, it's 11 cis, and then it changes its structure to an isomer where you no longer see it bent there. Now it's called all trans. So this activates a protein, right? That activates an enzyme. Here it says enzyme, it ends in ACE. It activates second messengers like cyclic guanosine monophosphate to eventually not to open channels in the membrane, but to close channels in the membrane. Okay. And you may ask why I go through this whole cascade. Well, this is called an indirect effect. Indirect effects take a while, but they're longer lasting. So you're not just closing one channel, you're closing a bunch of channels in the membrane. So instead of, let me go ahead and erase this see it a little better. So here, normally the channel's open. You'll notice sodium ions coming in. And then when the channel closes, no longer are sodium ions coming in. So this is a little bit backwards. Most often when you excite targets through neurons, you're trying to depolarize them, make them more positive inside. But here, you initiate the signals to the brain by hyperpolarizing the photoreceptor, not depolarizing it, hyperpolarizing it, which means you're making it more negative inside. I know it's a little bit backwards. So here they give us two scenarios, one in the dark, one in the light. So let's have a look what's going on in the dark. So here we got our photoreceptors, and you're going to notice what is the resting membrane potential. The resting membrane potential is not at minus 70 millivolts. It's starting at minus 40 millivolts. So it's kind of already in a depolarized state. And when it is depolarized, neurotransmitter is constantly being released from the photoreceptor here. And when it releases neurotransmitter onto the next cell called the bipolar cell, if neurotransmitter is being released, what does it tell the bipolar cell to do? It tells it to make it more negative inside. It's hyperpolarizing it. And when it hyperpolarizes the bipolar cell, it doesn't send signals to the next cell in line, which is the ganglion cell. Okay. And those ganglion cells, if they get activated, are the ones that make action potentials to send signals to the brain through the optic nerve. Okay, so let's go and erase some of this. So now let's see what happens. Light does hit the photoreceptor, and now instead of depolarizing it, it's hyperpolarizing it. So hyperpolarizing the photoreceptors causes the receptor to no longer release neurotransmitter. Therefore, that activates the bipolar cell to go ahead and release neurotransmitter onto the ganglion cell, therefore creating action potentials, which are signals that you're going to send eventually to the brain to interpret what you're seeing. So this little picture here is just showing us that when you stimulate photoreceptors, you're not depolarizing them with light, you're actually hyperpolarizing them. Okay. We're not going to go into too detail here. The only things I want you to take note is that the optic nerve, some of the axons will decussate, which means cross over in an area called the optic chiasma or chiasma. And then the information goes to your relay center. The relay for any sensory, remember, is going to be your thalamus. So somewhere in here, they're going to label thalamus right here. So the thalamus is going to relay the information next to the cortex located in your occipital lobe to interpret what it is you're seeing. And your optic nerve is cranial nerve numeral 2. Clear this drawing out. All right, let's go to the next picture. Yeah, it's just a brain here. You could see here that the optic nerves from each eye have been cut. So here, these are optic nerves that eventually connect to each eye. And then some of the axons, remember, will decussate right here in the area called the optic chiasma. Well, that's cranial nerve numeral 2. It's purely sensory, which means you're only sending information to the brain. Cranial nerve numeral 1 is called olfactory. Comes off this bulbous region here. Olfaction is to interpret what you're smelling. Only those two nerves, cranial nerve 1 and 2, are purely sensory, where you're only sending information to the brain, not from the brain to a target. Okay, now we're leaving vision. We're going to look at cranial nerve numeral 1, olfactory, right? So cranial nerve numeral 1 comes off a bulbous region called the olfactory bulb. And this is the cranial nerve number 1, which goes through your ethmoid bone. So this is the area here. It says cribriform plate of the ethmoid bone. So here these neurons form cranial nerve numeral 1, olfactory. You do make mucus. You have some support cells here that make mucus. The mucus is going to help to trap the chemicals that are traveling in the air. They travel in the form of a gas, right? And the mucus helps to trap the chemicals in the gas, right? So that gives time to stimulate the receptors to interpret what it is you're smelling. And notice these olfactory neurons, they have cilia, not to necessarily propel mucus here. That's not their function. The cilia here is to increase surface area, kind of like your fingers branch out so you can grasp objects. So in this case, you're increasing surface area so that the chemicals can stimulate the olfactory cells. And this picture is just kind of iterating what the book wants you to know is that because the receptors are up high, it may take you to inhale or sniff gases here to stimulate the receptors for smell. Almost said photoreceptors, no. The ones for smell are olfactory. Um, excuse me. One of the experiments in lab, get a chance to do it, is you're going to have some taste tests. Well, you'll be given a candy and you'll be asked to distinguish the flavor. Now, the person you're testing, excuse me, you're going to ask them to hold their nose, and they should not be able to detect the flavor right away. You'll note that smell plays a good role in distinguishing certain tastes. So smell plays about a good role, about 80 plus percent of distinguishing different types of flavor. So if I can't get chemicals to stimulate these receptors by holding the nose for a short period of time, I won't be able to distinguish the flavor of whatever I'm putting in my mouth. Then you'll ask the person to open their nose. Then they should be able to distinguish what flavor of candy they have in their mouth. Excuse me. So in times where maybe you're have congestion of your nasal sinuses, during when you have colds, some of the chemicals here are obscured. They're not being able to stimulate the receptors. You'll have a more difficult time interpreting what it is you're tasting. Okay. Now, again, if you allow too much time for the individual while they hold their nose, some of the chemicals will be broken down, right, the food or whatnot by saliva, and some of the chemicals can travel up from the back to stimulate the receptors for smell. So when you test someone by giving them the candy, make them look away so they don't know what flavor candy you're giving them. Two, ask them within a couple seconds, yes or no, can you distinguish the flavor? Don't wait too long because, again, some of the chemicals can travel up from the oral cavity into the nasal cavity from the back. So again, you have the olfactory nerves, and then you got support cells here making mucus. And just to take note, there's different types of neurons. That was in chapter 11. You have multipolar, bipolar, unipolar, where you look at the cell body and you see how many processes come off the cell body. So if it looks like if there's only two processes coming off the cell body, like in this case for olfactory, this is a type of bipolar neuron, bi indicating two processes coming off the cell body. But regardless, they're all going to have an area where they can receive chemicals or messages in the form of like neurotransmitters, or in this case, gases. And then they're going to have the end of the tail where they can send neurotransmitters to communicate to other cells. And you'll notice a nerve, cranial nerve numeral 1, olfactory, is coming off the brain or spinal cord. And once you enter this bulbous region, the axons are no longer a nerve. You refer to axons once you're in the brain or spinal cord as a tract. So that's why it's referred to as tract here. Okay, so let's have a look here. The chemical traveling in the atmosphere stimulates, or well, actually binds first to a protein receptor, activates second messenger proteins to activate an enzyme, which eventually will cause the opening up of chemically gated channels to allow sodium to come in. So yes, chemicals in this case do depolarize the target for the receptors interpreting smell. Now going to taste. Let me get a larger picture here. So there's actually five submodalities of taste, or categories you can give the sensation of tasting. items that are sweet, which is more you have the taste buds, which is an organ on the tip of the tongue for sweet. Sour, salty, you tend to indicate those on the side of the tongue, and bitter toward the back. And then there's another taste sensation that Japanese got to discover it, so they got to name it. It's umami, which gives you the sensation of like protein or things like a cheese wise that your book says that umami. So it's five submodalities of taste, but don't forget smell plays a good role in distinguishing different flavors, right? Without smell, you wouldn't be able to taste nearly as well. The other things that play a role in certain tastes, there's maybe like texture of food, temperature of food as well. Even spicy or spices that we use tend to stimulate pain receptors, also play a role in taste. Hmm. Okay, let me move on. So here you're looking at a taste bud, and within the organ, because taste bud is an organ, remember, is two or more tissues. Within the taste bud are the receptors here to interpret the taste that you're sensing. The other thing I wanted to mention, oh, well, this picture illustrates it well, is that the receptors that interpret taste are called gustatory. Gustation means taste, and they have these extensions called gustatory hairs. And because friction occurs in the mouth, right, these are constantly being broken down and regenerated. So you're constantly regenerating these gustatory hairs. And then the pore is just an opening to allow like chemicals to enter to stimulate the gustatory receptors. These are the cranial nerves involved with sending information to the brain on what you're tasting, the sensation of taste. So which cranial nerves are involved? It looks to be cranial nerve. Our friend vagus, number 10, can also send sensory to the brain with taste. Cranial nerve 9, glossopharyngeal, and cranial nerve numeral 7. They all send information to the brain to interpret sensations of taste. And if you recollect, there's three pathways, three neurons when you send sensory up to the brain. The first pathway is always where the receptor is stimulated. Then you got a second pathway going up, and then the third pathway is always in the thalamus, our relay for sensory, and therefore it's going to relay it to the appropriate cortex to interpret what you're tasting, and it's called the gustatory cortex, located in an area called the insula. All right, let's go to hearing. Now let's get to a larger picture here. So the outer ear itself is referred to as auricle or pinna, and the way it's shaped helps to gather sound, and sound travels in waves. Okay. So the sound's going to enter and be gathered up by the ear, the auricle part. And this portion inside here is called the external acoustic meatus. And then here, when you get in here, is the internal ear. Let's have a look here. So here, you commonly probably know this as your eardrum, but it's properly named as your tympanic membrane. And it's funnel shaped. So that's going to help to gather the sound waves inside. And the sound waves are gonna cross over through these bones here. The bones are in general called ossicles. You have three of them. The first one's name is malleus, second one's name is incus, and the third one's name is stapes. And the job of the ossicles, these bones, is to provide like a bridge so that the sound waves can get to the organ where the not photoreceptor, where the receptors are to interpret what you're hearing. Okay, so one, the ossicles act as a bridge, and two, the ossicles amplify the sound waves so that you can interpret what you're hearing as being louder. How do they amplify sound? Well, if you notice here, the ossicle here comes to a fine point. Anything where you can focus something at a fine point, you're amplifying it. Think of, let's say, if I want to amplify your weight, you could wear high heels, and at the end of the heel, your weight was amplified. Vice versa, if I want to distribute your weight, I would wear snow boots where it's like spreading out your weight so you don't sink under the snow. So where it comes to a fine point here, Another job of the ossicles is to amplify sound so you can interpret what you're hearing as being louder. And then the receptors to interpret what you're hearing, they're located within this organ here. And it kind of looks like the shell of a snail. And this organ that has the receptors to interpret what you're hearing is called the cochlea. And I don't see it labeled here. And then continuous with the cochlea, you have this organ as well attached to it called the vestibule. And then from the vestibule, you have these circular structures called semicircular canals. So in the vestibule, you have receptors to interpret balance. Okay. And then the information for hearing and balance is conveyed through these two nerves. One from the cochlea, it would be conveying what hearing, what the sound waves are interpreting what you hear, and then here for balance. So these two nerves come together. It forms cranial nerve numeral 8. It's called vestibulocochlear nerve. And I'm writing the Roman numeral for 8. Vestibulo represents the vestibule. Cochlear represents the cochlea. So this nerve sends signals to the brain, not just for hearing, but also for balance. You'll notice as well in here, this is the stapes, that ossicle, and it lies against an area where sound waves are going to be able to go to the cochlea, and then there's an opening here referred to as, I believe, the round window that allows an exit for the sound waves. And then continuous here, this leads here down to your pharynx region above your oral cavity, okay? And this tube is called your auditory tube, also known as pharyngotympanic tube. The function of this tube is to equalize pressure so that sound waves can properly go to the cochlea. Usually, typically, I'll tell students, if you've ever been in an aircraft, there's a difference in pressure as you start going up in altitude than what's trapped in your ear. So as pressure changes around your body, sounds start to sound muffled. They don't sound as loud because there's a pressure difference. But as you start moving your mandible, like if you were chewing, air will equalize through the auditory tube and you'll hear like a little popping sensation. And that's when the pressure inside the ear is now equal to what's outside the ear. And now sound waves can properly go to the cochlea and you hear just fine. And similar again in the aircraft, when you start going to land, you'll feel the pressure differences occur again as you're descending in altitude as well. So the job of this auditory tube is to make sure the pressure is the same inside the ear canal as it is outside so you can hear it properly. Oh, much better picture here. So here we got the tympanic tube, which you commonly know as your eardrum. Here you got the ossicles. The first ossicle's name is malleus, which means hammer. Second ossicle's name is incus, which means anvil. And then the third ossicle's name is stapes, which means the stirrup. So I don't know why students always tend to call it hammer or anvil, but no, when the lab practical comes, you'll want to properly name them malleus, incus, stapes. This here is referring to like a blacksmith who uses a hammer to shape metal on an anvil and maybe pulls or steps on a stirrup to increase the flames as he's shaping the metal. Okay. Now, sound waves are going to enter this area here that the stapes sits upon. This is referred to as the oval window. And then sound waves enter the cochlea. That's the organ that has the receptors to interpret what you're hearing. And then once it stimulates the receptors, there's an exit window here called the round window to allow the sound waves to exit. And then the nerves, the nerve leaving the cochlea is called cochlear nerve. And then you have this organ called the vestibule, and that's going to give you a sensation of balance. And then also you have semicircular canals to give you a sensation of balance as well. One gives you a sensation of what's called static equilibrium. In other words, giving a sensation like going up, down, side to side. And the semicircular canals will give you a sensation of what's called dynamic equilibrium, which basically tells your brain where your head is as you're rotating it in an angular movement. So balance is going to be conveyed through this nerve called the vestibular nerve. And eventually these two nerves come together and they form cranial nerve numeral 8 together called vestibular cochlear nerve. There's the Roman numeral for 8. Now, cranial nerve numeral 8 was once thought to be purely sensory, where you only send information to the brain. The only nerves that are purely sensory, again, are cranial nerve numeral 1, olfactory for smell, and cranial nerve numeral 2, optic for vision. But once thought cranial nerve 8 was purely sensory. However, you do send some information motor to communicate to these muscles here. Okay. So if you're in a loud environment, you don't want to over amplify sound and cause damage to the receptors for sound inside the cochlea. So in times when you're in a loud environment, you do communicate to these muscles here that can help separate the ossicles a little bit so you don't over amplify sound, causing damage to the receptors for hearing. So yes, cranial nerve numeral 8 does have a little bit of motor, which means communications to areas in that in here in the ear canal, communicating to these muscles. All right, we're looking at the receptors here for balance. Balance, the receptors again are located in the vestibule and also in these structures called semicircular canals. And they give you the sensation of static equilibrium, which is going up, down, side to side. And then the other receptors here are for dynamic equilibrium, give you the sensation of where your head is as you're rotating it. I like to think of maybe like a shampoo commercial where the model's waving their head, right? And as they wave their head, your brain knows where your head is as you're rotating it. That's called dynamic equilibrium. Now, if I were able to cut into that organ that has receptors to interpret what you're hearing, it kind of, you're going to see it here. You cut into it, and you have these canals here filled with fluid. So let's magnify that in one of these canals. So in the middle canal, it's called the, get the pen out. It's called the cochlear duct. And the fluid in here is the deepest, so it's referred to as endolymph. And then you got these two canals above and below it called scala. What do we have here? Scala vestibuli and scala tympani. And the fluid inside these here is called perilymph. Peri, that prefix peri, means to surround. Okay, so now in the next slide, they're going to magnify this cochlear duct that has the receptors here to interpret what we're hearing. Here we go. So here are the receptors to interpret sound. And the way they get stimulated is as sound comes, it displaces fluid. And when fluid's displaced, it moves this membrane called the tictorial membrane. And as the tictorial membrane moves, it's moving these little hair projections that are coming off the receptors for hearing. So as sound displaces the membrane, you activate the receptors, they make action potentials, and then they send signals to the brain to interpret what you're hearing through cranial nerve 8 called vestibulocochlear nerve. So an actual, I guess it's an electron microscope picture here of the hair cells of the receptors to interpret what you're hearing. And again, sound travels in wavelengths, okay? So here's, you may have an experiment in the lab where you'll test the individual's ability to hear by hitting this tuning fork, which will create sound waves, and you'll move it behind the individual so they can't see, move it in different areas around the head and ask them where they hear the sound coming from. Okay. Now let me have a look at this. Now, we talked about frequency is something that occurs, and the more times it occurs, we say it has a higher frequency. So the wavelength in red has a higher frequency where it occurs more often per time. So sounds that have a high frequency, you interpret that as what's called a high pitch. Vice versa, if sounds have lower frequencies where their wavelengths don't occur that often per time, you interpret that sound as being a low pitch. Now, sound can be amplified by the ossicles. Amplify means you make the peak of the sound wave go higher. So sound waves that have a higher peak, you've increased its amplitude. So sounds with a high amplitude, you interpret the sound as being louder versus sounds with a low amplitude, you interpret as a softer sound. All right, let me get to a larger picture. So sound is going to enter the ear canal, right? Gets funneled in through the tympanic membrane, and then it is going to be allowed to cross over through the ossicles here. And not only that, it's going to be amplified, and then it's going to travel within the cochlea and stimulate receptors located in this duct called the cochlear duct. So if they could unwind the cochlea where it looked like a shell, this is what they're doing. They're unwinding it in the picture. So you could see the different ducts here. You got scala tympani, scala vestibuli, and in the middle where the receptors are located, that duct is called cochlear duct. This picture is showing us that sound, depending on the frequency, sounds with a high frequency will activate the receptors near the base of the cochlear duct. Whereas sound with lower frequencies, right, they will activate receptors near the peak, the apex of the cochlear duct. All right, so here's some of the receptors. I imagine these were what? The ones to interpret sound in electron micrograph picture. Now, which cranial nerves are involved in sending information as sound? Well, we said cranial nerve numeral what? 8, which would be your vestibulocochlear nerve. So once you stimulate the little hairs on the receptors, they'll make action potentials. And through cranial nerve 8, you'll send information to the spinal cord, right? And there's 3 pathways. So the first pathway is the receptors that get activated, second pathway in the spinal cord, and then the third pathway is always in your relay center, which is your thalamus. Okay. And then thalamus will relay the sensory to the temporal lobe where you have cortex to interpret what you're hearing. I did want to mention, I guess, let me jump back. We do make earwax. You have glands in the ear canal to make earwax that are there to help prevent insects from making housing in the ear. But sometimes you may have so much earwax that it blocks or obstructs the sound waves to going where they need to go so you can hear well. So if that's the case where you have accumulation of earwax, you're blocking sound waves, that's called conductive deafness. You're blocking the conductance, which is the movement of sound waves to where they need to go. So conductive deafness, blocking, okay, blockage like earwax. And that's where you'll get like, well, I've had that done where you get liquid or whatnot from the doctor to break the earwax and then they flush it out with warm water and a syringe. Now, what you don't want, you don't want deafness at all, but you don't want something that's permanent. All right. So if you have damage to the nerve that conveys information of sound to the brain, that's called nerve deafness. Okay. All right. Now let's go back to where we left off. Okay, so let's look at the receptors to interpret, balance now. So in this organ here called the vestibule, you have the receptors to interpret static equilibrium. Here's the receptors, and they're embedded in a membrane called the otolithic membrane. I like to think of maybe like Jello, right? It's like Jello where the receptors are embedded in the Jello, in the otolithic membrane. And you'll notice that one's oriented vertically, so that gives you the sensations of going up, down. As you're moving, you're displacing the membrane. So if you displace this membrane where it's vertical, that gives you a sensation of going up and down. Vice versa, you also have receptors embedded in an otolithic membrane that's horizontal. So that way, when the otolithic membrane gets displaced, that gives you the sensation of going side to side. So this type of balance where you feel like going up, down, side to side is called static equilibrium. Okay, now you also have receptors here in the semicircular canals. And when you rotate your head in an angular movement, that tells the brain where your head is. That type of balance, that's referred to as dynamic equilibrium. That's the scenario where I think of the shampoo commercial and the models moving their head in an angular movement to help me remember dynamic equilibrium. So here's just showing you as you move your head angle-wise in different areas, you're stimulating the receptors in those semicircular canals, giving sensations of what's called dynamic equilibrium. So depending on where your head is rotated, you may make more action potentials or you may make less. And then here you're just seeing the receptors, they are located in the semicircular canals. It looks like they're also embedded within this, what's called ampullary cupula. And this next picture, let me see if there's a larger one. No, there's not. Basically, to give you the example of dynamic equilibrium, this ice skater is rotating, rotating, rotating. Their head is rotating, right? And as their head rotates, you displace some of the fluid here that physically moves the receptors for balance, creating action potentials that send information to the brain where your head is as you're rotating. And then here's an electron microscope view of these receptors located inside this semicircular canals. I think that's pretty much it for this chapter 15, the last chapter on sensory. Oh, not quite. Let me just emphasize something before I let y'all go. If you remember the job of your cerebellum, the cerebellum is to provide a map of how to get from point A to point B smoothly. It constantly knows where you are as you're moving. Yes, you send information to the brain as you're moving as well, but for the cerebellum to provide a map how to get from point A to point B smoothly, it has to receive information as you're moving. So it's getting information visually, it's getting information balance wise, and it's getting information from receptors in your muscles as you're using your muscles in movement. So you send all this information, right, to the cerebellum, right? And through the cerebellum, you can execute smooth movements from point A to point B. And the key words in the chapter 12, the job of your cerebellum is responsible for smooth, coordinated skeletal muscle movements. So your cerebellum can allow you to respond quickly to different stimuli. All right, let me erase all that. And I think that should be it. Oh, not quite. Let's just emphasize some again. Rods, you have more rods than you do cones. The job of a rod is to amplify light when there's very little, right? So you use them at night. Cones, they're more to interpret color and for visual sharpness. So let me give you an example of how we use these. When you're ready to go to bed, you hit the lights, right? You can't quite see anything right away. First of all, you hit the lights, they're gone. Your cones are out of play. And you allow a little time for the rods to adjust to what little light there is. And then once they adjust, you start to make silhouettes out when the lights are off. You can see things in the room. Now, vice versa, let's say you're in a movie theater and you went from a dark environment immediately into a bright environment. When you go immediately from dark to light, what happens there is that when you get from dark to light, the rods are now overstimulated. They go out of play. You're blinking your eyes because you're not used to that much light no longer. You're stimulating the rods too much. And as the rods go out of play, what comes in play next are the cones when it's daylight. And again, you have more rods than you do cones. So it says here you have about 20 rods for every one cone you have. Rods are more for peripheral vision, whereas cones are more for central vision. Remember, the job of the lens is to focus light where there's nothing but cones, called the fovea centralis. Okay, for balance, for static equilibrium, that gives you sensation going side to side or up and down, right? It's called static equilibrium. The receptors are in what's called the organ called the vestibule. For dynamic equilibrium, where your head's rotating in an angular movement, that's called dynamic equilibrium. The receptors are located in an organ called semicircular canals. And then the receptors to interpret for what you're hearing are located within an organ called the cochlea. All right, that's it. I don't know how this picture here pertains to the sensory, but we'll just leave it at that. And we're done with chapter 15. And that should be it. I'll see you around soon.